The first step in determining if a mutant protein was created through either an addition or deletion of a base pair in the normal mRNA sequence is to observe the amino acid sequence in the original and mutant proteins. After noting the amino acid sequence, you need to determine all of the possible codons that could result in the production of each amino acid, and then proceed to write those in sequential order. To do this, you need to view a codon chart. Here I have listed all of the possible codon sequences for the normal mRNA sequence, using superscripts to designate situations in which a specific codon could utilize either of the listed nucleotides to code for an amino acid, and underscores to designate situations in which any nucleotide, adenine, cytosine, guanine, or uracil, could be used in that specific position to code for the same amino acid. Now I have listed all of the possible codon sequences for the mutant mRNA sequence, using the same format to designate the potential possibilities of nucleotide placement. The codon UGA was placed underneath UA superscript AG to avoid confusion because it is another potential stop codon, but the two do not share an identical second nucleotide. Next, you need to examine the first position of each codon in the normal mRNA sequence. I have circled each letter accordingly in red. As you can see, there is agreement within the two sequences' first letters up until the third amino acid, where there is the first discrepancy. This is what will help us determine if the mutation was caused by an addition or deletion, and what the exact codon sequence must be of the normal mRNA and mutant mRNA. As you can see, there is no adenine nucleotide in an adjacent position respectively in the normal mRNA sequence. This is our first clue that the mutation was caused by an insertion rather than a deletion. To determine this with certainty, we must now examine how the nucleotides in the mutant mRNA shifted in comparison to the normal mRNA after the addition of this adenine nucleotide. The nucleotides after this adenine nucleotide should have shifted to the right if there was an insertion. Therefore, each of the initial nucleotides that were in the first position of each codon should now be in the second position in the respective mutant mRNA codons. As you can see, this is the case. In addition, each of the nucleotides in the second positions of the normal mRNA codons are in the third positions of the respective mutant mRNA codons, which have been circled in blue. The nucleotides in the third positions of the normal mRNA codons are now in the first positions of the next adjacent codons of the mutant mRNA, circled in green. The addition of one base pair in the first position of the third codon in the normal mRNA sequence, shown in purple, resulted in the formation of this mutant mRNA. This resulted in a nonsense mutation, because the mutant mRNA was cut short after the last nucleotide in threonine was shifted to the right, creating the codon UAA, which is a stop codon. However, the last nucleotide in the codon coding for lysine and glycine cannot be determined definitively due to the mutant mRNA resulting in a nonsense mutation.